What I have here is an ear that's been infected with Gibberella ear rot. And you can see at the tip of this ear, it's kind of a whitish, pinkish type mold that starts at the tip and works its way down. Once in a while in the field you can see this where, depending on the different hybrid that you're working with, it can start at the base and work its way up a little bit, but that's just on certain hybrids, and most of those are hybrids that kind of dry down with the ear in the upright position. Most of the time though, when we're scouting fields, when we see this, it's at the tip of the ear, and that's definitely the most prevalent. Why the tip? This fungus enters the ears through the silks. Okay? These spores are basically hitchhikers. Okay? They jump on that pollen and ride that into the ovaries, and that's where they affect they develop, and that's where they produce the mycotoxins within that ear. Okay? So when you have weather out there in the summertime during silking, weather that favors silk elongation and pollen shed, that's also weather that favors the spore development and infection of these ears. So how do we manage it? Really, there's three management things that we're going to talk about here today. The first one is hybrid selection, and by far, if you can find some hybrids that are resistant or offer some resistance to Gibberella ear rot, that's definitely going to be your best option. The problem is there's not a whole lot of that, and sometimes getting that data from your seed companies can be kind of tough. The other option is crop rotation and residue management. The problem with crop rotation is the same fungus that affects the corn in the ear rot also affects soybean seedling disease and gives us those seedling diseases. It also affects wheat in terms of head scab. And then in corn we have the ear rot and the stalk rot. So rotating to a different crop really is not going to resolve that issue. As that crop dies and that residue is right there on the soil surface, in the absence of tillage, there's no way to destroy that residue, and those spores are just hanging out there on the top of the soil waiting for the next crop the following year. So that's why this one may not work as well. Another option that we have is the use of fungicides. In most of the research trials, fungicide use has been highly variable. And the main reason for that is just because of some equipment limitations. If you think about it, if you have to go in and spray a fungicide, around silking time when that plant's, tasseling, that's some pretty tall corn to drive through, okay? So not only are you driving through tall corn, but you have to get that fungicide down around the ear on that plant. So you have to have some sort of drops or some kind of way of getting that fungicide to the right part of that plant. So as you're driving through there, you've got drops on. If you're on a nice level field, that's one thing. But if you're in a field that has some rolling terrain or some of the hillsides like we have in Knox County, that can be pretty tough. And I think that those application issues are why we're seeing some trouble with fungicides not being as consistent for control. What if we had weather data that we could use to predict vomitoxin levels in your corn? Right now, for those of you guys who grow wheat, you're probably familiar with the head scab model that Dr. Pierce Paula, our plant pathologist at Worcester, came up with. That does a pretty good job of predicting head scab levels by looking at existing weather data. Currently, he's trying to work on the same sort of model to predict vomitoxin levels in corn. Basically, what he's looking at is rainfall, temperature data, surface wetness, those types of things on corn plants, basically at silking, and 5 days on either side of silking, 10 days on either side of silking, or 15 days on either side of silking. And what he's come up with, a model that he's currently working on, it looks like that period 7 to 21 days after silking seems to be the most critical period. And within that time frame, you're looking at temperatures between 59 and 68 degrees and relative humidity over 80%. Okay, and how many hours each day those plants are affected to those conditions. And with that model, he's been able to predict vomitoxin levels within 1 part per million with 80% accuracy. Now think about that for a minute. If you can predict based on weather patterns with an 80% accuracy or better, the chance of you having vomitoxin in your corn this year, I think that's something that we'd all like to have, whether we're a grain farmer, whether we're a livestock farmer. Those would give us some options to make some different plans throughout that growing season to help us with that. Whether that's some sort of a rescue treatment with a fungicide, whether that's segregating our corn in different bins, whether that's maybe looking forward to buying some different feed for our livestock, it'll give us some different options down the road. And I think that's really important. Again, this is something that's under development right now. So hopefully in a few years, we're going to be able to come up with a model where we can just look at weather data and be able to predict how much vomitoxin is going to be in our corn.
As John talked about, Gibberella ear rot is the main concern when we're talking about vomitoxin. Vomitoxin is a mycotoxin. There's over 200 different mycotoxins, but vomitoxin is one of them that is specifically produced by the Gibberella ear rot. And it favors that alternating warm and cool temperatures during that growing period, the same ones that favor pollination. So what happens with livestock when they're fed corn that's contaminated with vomitoxin? Normally there's a sharp decrease in feed intake, which in turn lowers that growth rate. If we feed corn that has the mycotoxin, vomitoxin, to animals for a long period of time, we can have vomiting and diarrhea, as well as digestive lesions. Also, sudden death may occur. The good news is there are some binder products out there that will bind the vomitoxin. However, we want to be sure we're working with our feed mill when we have these issues, because some of these binders will also bind thiamine, which may also need to be added into the diet. So when we talk about vomitoxin and we look at other feedstuffs, some of us may be hauling our corn to ethanol plants where DDGs are the byproduct. Those DDGs will be fed to livestock eventually. When we look at DDGs, they actually come in at 3 times the rate of vomitoxin. So if your corn is testing at 8 parts per million, the DDGs will come out at 24 parts per million when we talk about vomitoxin, which is definitely not what we want. Swine are very highly sensitive to vomitoxin. Anything above 1.5 parts per million, they will start to decrease their feed. For every 1 point above 1.5 parts per million, they will decrease their feed intake by 4%. Ruminants are a little more resistant to this, which is why we can have some livestock that will eat some of this corn eventually. The solution, like we talked about, are binders, as well as you can dilute your corn. So if you find some good corn, some not so contaminated vomitoxin corn, you can dilute that out with your higher vomitoxin corn and have the solution. The one thing that we want to be sure we're all talking about is that vomitoxin is not the only toxin that is created when we have a Gibberella ear rot. We can also have a Mycotoxin Zoroalinone. Since it's produced by the same mold, it obviously has the same growing conditions, that alternating warm and cool through the growing season. This mycotoxin specifically affects reproductive traits. It has an estrogenic effect. So anything that you would think of feminization or anything to do with reproductive tracts is what is affected by Zearalenone. If we feed it at lower rates, we could have some prolapses, we could have anestrus, which is when those cows or sheep or goats or pigs aren't cycling, they're not having their normal reproductive cycle. We could have false pregnancies, and we could have embryo losses. That more prolonged period of time of having that fed to them, we could have poor viability of newborns and abortions. When we feed this to males, we can actually see some low libido, poor sperm development as well. Zearalenone actually does not have any FDA suggested guidelines. However, here are some guidelines that we do have from Purdue and the chemistry department of Indiana. We can see that at 0.5 parts per million, we have reproductive efficiency affected in gilts. So at 1 part per million, you can have some animal feminization. So if you're trying to run a boar stud, or if you're trying to have some male offspring to carry on for a stud for another flock or herd, this could affect some of that. And anywhere from that 50 to 100 parts per million area is when we see some conception and ovulation issues, along with fetal development and abortion problems. There's been limited efficacy on any binders that are out there. The ones we have seen are the aluminosilicates, which are those clays, but like we said, very limited efficacy. When we're talking about cattle, for developing heifers at 5 parts per million is when we can start to see some reducing conception rates. When we're at 10 parts per million in breeding cows, we start to see some issues with the reproductive cycle as well as conception rates. And at 20 parts per million is when we start to think and see some abortions and loss of fetuses. When we talk about swine, for those gilts, anywhere from 1 to 5 parts per million of Zearalenone is when we start to think and we start to have some issues with swelling, as well as those gilts not coming into their first cycle, so we can't get them bred. For sows, between 5 and 10 parts per million, we start seeing a little bit of latency in that post-weaning interval of estrus. So getting those sows back from their weaning back onto their reproductive cycle, we're starting to have some issues. Normally that could be around 3 days. With a 5 to 10 parts per million of zearalenone, we'll see that start to get very much longer. Above 25 parts per million, in gestating sows, we'll start to see a lot of issues with small litters and abortions as well. When we talk about boars, anywhere from 5 to 10 parts per million of zearalenone is when we're starting to see a lot of issues with sperm mobility. And then when we get above that 20 parts per million, we start to see a lot more issues, including small testicle size. Have you ever wondered what happens when they actually test your corn for vomitoxin? You're about to find out.
This year has certainly been different. We're hauling grain this year and you pull in there and a little bit the same, but then they pull you off the side to run the vom test. What goes into testing corn for vomitoxin? Here's a little bit of what it takes. When you pull in, it's much like every other year. They're going to try to get a good random sample out of the load. When they get the sample, they're going to test the corn for moisture and they're going to test it for test weight. Once they get the sample, they're going to separate it to even smaller, evenly distributed samples.
This here is a divider. It'll split the sample. So that's their divided out sample.
Then they're going to weigh out about 50 grams. This sample will be used in the vomitoxin test. Once they get the size of sample they need, they will take this sample and grind it into a coarse powder. The coarse powder is then placed in a specialized filter bag.
See, it's got a screen in there. See the screen?
Then a buffer bag, which is a packet of chemical, is placed in the filter bag along with the ground corn. They then add a specific amount of distilled water to the bag. The next step takes a little bit of time. The mixture is shook up for about 2 minutes to dissolve that buffer packet. Then the mixture has to rest for additional 2 minutes. You then take a very small sample of the fluid for testing. You take 50 microliters, which is 5/100ths of a milliliter. You place it in the mixing container. Then you add a dilution solution to the container, snap it up, and shake it again.
Basically what we're doing is we're diluting it with this dilution stuff. So we're sticking that in there. And we're shaking.
Now we have a solution that's ready for testing. A microcell container is placed in the incubation chamber. Then we take 100 microliters of the previous solution and add it into the microcell. This solution is mixed with another chemical to produce a chemical reaction. A test strip is added to the incubation chamber. This chamber is closed and it incubates for 3 minutes. After 3 minutes, the test strip is removed and verified that the test was run properly. The test strip is placed in the reader, the test barcode is inputted, and the correct test is selected. The reader analyzes the sample and gives us a vomitoxin parts per million results. In this test, our results came up 3.91 parts per million.